to show that more transpiration occurs from the lower surface of a leaf. Take a freshly plucked leaf from a dicot plant. Dry 2 cobalt chloride paper strips over a flame. They will turn blue in color. Place the dried blue strips, one on the upper surface and one on the lower surface of the leaf. Cover both the paper strips with glass slides and observe carefully. It will be seen that the blue cobalt chloride paper on the lower surface of the leaf turns pink faster than that on the upper surface. This shows that more transpiration occurs from the lower surface of a dicot leaf. To show that green plants prepare food. Procedure. Take a healthy potted plant with fully green leaves and keep it in sunlight for several hours. Pluck a leaf and test it for starch with the help of iodine. The leaf turns blue-black in the presence of iodine, showing the presence of starch in the leaf. This shows that starch was prepared by the green plant during photosynthesis. To study the different parts of a typical flower. Take a large red-colored hibiscus flower. This flower has all the floral holes. Observe the floral holes one by one. The flower contains sepals, 5 in number, all fused to each other, green in color. Petals, 5 in number, all free from each other, red in color. Stamens. Numerous stamens are present but all fused to form one long tube. Anthers are yellow in color, filament is red. Carpels. 5 in number, all fused. Stigma is visible from outside, but to see the style and the ovary, we have to cut open the flower. All the holes are arranged on the thalamus, which is present on the tip of the pedicel of the flower.
Study of dicot and monocot seed. Soak some gram or bean seeds and maize grains in water overnight. In the next morning, take them out and observe them externally. Then open the swollen seed coat and observe the internal structure. Make a diagram of both the seeds in your exercise book. To study the 2 types of germination. Take some bean seeds and some maize grains and soak them in water overnight. Prepare 2 pots with healthy manured soil. Next morning, take out the seeds and sow the 2 types of seeds in 2 different pots. Place the seeds about half an inch below the soil surface. Water the pots regularly and observe them carefully. Note your observations after a few days. Draw diagrams of what you have seen. To show that suitable temperature is needed for germination, take 3 petri dishes and place wet cotton wool in them. Soak some seeds and then place equal number of seeds in all the 3 dishes. Keep the first petri dish inside a refrigerator, second one inside a room, and the third one on an oven. Note, where germination occurs, it will be seen that germination occurs only in the second petri dish kept in the room, since the seeds receive an optimum temperature necessary for germination. To show that water and oxygen are needed for germination, take a beaker and half fill it with water. Take a glass slide and tie 3 soaked seeds A, B, C at 3 positions as shown in the diagram. Place the glass slide inside the water of the beaker in such a way that seed A is completely submerged in water. Seed B is half in air and half in water, and seed C is completely outside water. Keep this setup at room temperature for a few days. It will be seen that only seed B germinates as it receives all the conditions necessary for germination.